Welcome back. This is Lecturette 2, where we will review the gross and microscopic anatomy of skeletal muscle. Now, the smallest unit of skeletal muscle that is alive is a muscle fiber, aka a muscle cell. And if you remember from Lecturette 1, it's called the muscle cell is called the muscle fiber because it is very, very long. Now, all muscle cells have connective tissue that surround them. And it allows you now to have or to bundle up a whole bunch of muscle fibers together. So this bundle of muscle fibers is called a fascicle. And you folks can see fascicles with the naked eye. So if you cook meat low and slow, and then you're able to pull that meat apart, and those little tiny strings you see, those are yummy, yummy, yummy fascicles. Now, when you have many fascicles bundled up by even more connective tissue, that is now going to form the muscle organ. Okay. And so there's also connective tissue that surrounds that. So all the connective tissue collectively that surrounds the muscle fibers, the fascicles, and the muscle organ all come together to form the tendon that attaches skeletal muscle to bone. Now let's review the gross anatomy of skeletal muscle. So the overall function of skeletal muscle is movement. We contract the muscle organ, which causes it to shorten, which allows us to pull on and therefore move bone. And in order to move around, skeletal muscle needs to be able to both contract and relax. Also, skeletal muscle is an excitable tissue, meaning that its function is stimulated by electrical impulses. So movement is initiated from the CNS. So it needs to receive an electrical impulse in order for contraction to happen. And then the flip side of that is that relaxation occurs in the absence of an electrical signal from the CNS. Now let's review the microscopic anatomy of a muscle cell. The anatomy of a skeletal muscle cell can be confusing because everything looks like a tube and there are tubes within tubes within tubes. So I know that this picture is a muscle cell, aka muscle fiber, because here I see a nucleus. So I know that I'm at the cellular level. So here's your anatomy review. Structure 1 is the sarcolemma. Sarco means flesh, lemma means shell, and it is the plasma membrane of the muscle fiber. Now, these yellow tubes, labeled number 2, are called transverse tubules, aka T-tubules, where they are responsible for transmitting electrical impulses down deep into the cell. These blue bags, labeled 3, represent the sarcoplasmic reticulum, which are responsible for storing intracellular calcium. And when the electrical impulses from the T-tubules reach the sarcoplasmic reticulum, calcium can be released. Now, muscle fibers, aka muscle cells, are made up of these tiny tubes called myofibrils, which is labeled number 4. Myo means muscle, and fibrils are small, tiny fibers, okay? Now, of course, myofibrils are made up of even smaller proteins called thick filaments, which we will call myosin, and myosin have these paddles, and thin filaments, which we will call actin, and actin has a binding site for myosin heads to attach to. Now let's look at a myofibril more closely. Now, if you remember from anatomy, all the myosin proteins are bound together on this backbone called an M-line. And the actin proteins are bound together on this backbone called the Z-disc. Now, the goal of this anatomy review is not to rememorize all the names of all these lines, bands, and discs. So stop, drop, and roll if you're starting to get unpleasurable flashbacks from anatomy. The goal of this slide is to refresh your memory on how things are built, on the structure of these contractile proteins. So all the myosin proteins are bound by a backbone, much like how my arms are bound to my body. and I can move my arms to do work. But my backbone will function as an anchor point for my arms, which is the same for the anchor point of the actin and the myosin proteins. That's the take-home message, okay? Now, this section here that I'm circling, that consists of overlapping actin and myosin proteins, is called a sarcomere, which is the contractile unit of a myofibril. And the myofibril is made up of gazillions of repeating sarcomeres. Now, during contraction, the myosin head starts to paddle into actin, much like a kayaker or a paddle boater paddles, okay? Now here, look at our analogy. First, you have to dip the paddle in the water, okay? So our myosin head is the paddle and the water is the actin, okay? Then you have to pull the paddle back behind you, which pushes the water back, allowing the paddler to move forward. Then you have to lift the paddle out of the water and return it to the front of you and repeat, which allows you to now move forward. So when each sarcomere contracts, the sarcomere shortens because actin and myosin are sliding past one another, okay? So they're not changing length, they're just sliding past one another. Much like how a boater will slide or glide across the water. So this sarcomere here shows the muscle in its relaxed state. There's a big old gap here. But when myosin paddles into actin, the sarcomere shortens as actin, the water, slides past myosin, which is the paddle. All right, that's it for our lecturette 2. Next, we're going to start to review the neuronal control of skeletal muscle.